Hello, my name is Charles Carr, and today I'd like to speak to you a little bit about our investigation of the microbiota of presumed aseptic hip and knee implants. All of these implants were originally placed through surgeries called total hip or total knee arthroplasties, which are replacements of the components of these joints. Unfortunately, though, about 12% of these implants will require what's called revision or replacement. This is for a variety of reasons, including fracture, instability, infection, and what's called aseptic loosening. Aseptic loosening refers to failure that's not associated with physical injury like a fracture, and is not associated with detectable microorganisms either. Recently, though, several investigations have found that presumed aseptic implants actually contain detectable bacteria. This leads to our hypothesis that some cases of presumed aseptic loosening might actually be driven by polymicrobial communities associated with the implant. Some of these prior studies have utilized 16S rRNA gene sequencing to assess these bacteria, but we also hypothesize that this technique might be limited in this context. More specifically, we knew that DNA sequencing results can vary depending on the extraction methodology utilized. As well, samples with low microbial abundance, such as these, are more affected by contamination. And finally, several taxa that are very important in the context of hip and knee implants, like Cutibacterium acnes, are amplified especially inefficiently by 16S rRNA primers. To assess these hypotheses, we collected samples from the hip or knee implants of 41 patients requiring revision for presumed aseptic failure. Some of these samples were subjected to microbial culture, as a standard practice in the hospital to detect infections that were previously unknown. Specific for this study, some samples were subjected to DNA extraction, either with the PowerSoil high throughput kit or with the CTAB methodology. Extracted DNA was then used in 16S rRNA gene sequencing, as well as PCR specific for a few taxa of interest, namely C. acnes and Staphylococcus aureus. Using these approaches, we identified a positive control-associated artifact, in which some samples showed inflated proportional abundance of S. aureus and E. coli, the positive controls. The affected samples appeared in clear patterns in the 96-well plate layouts we used to prepare the samples for sequencing. The patterns largely correspond to the left and right primers we use for the positive controls, with only one exception. To verify that these patterns actually represented an artifact, we used decontam, which indicated that E. coli was a contaminant, and specific PCR for S. aureus, which excluded its presence in these samples that apparently had a very high proportional abundance of S. aureus. Unfortunately, this artifact was resistant to standard filtering procedures, so we used Source Tracker, trained on the open-air controls and positive controls, to identify a subset of samples that were relatively contaminant and artifact-free that we used for our subsequent analyses. Using this subset of samples, we determined that DNA extraction methodology had a significant influence on the overall observed diversity after sequencing. Specifically, the CTAB extracted samples showed a higher Shannon diversity than the PowerSoil extracted samples. We also identified several taxa that were significantly differentially abundant between these two groups. The PowerSoil sample showed higher abundance of several known laboratory and sequencing contaminants, whereas the CTAB extracted sample showed higher abundance of several taxa that might plausibly be part of the microbiota of presumed aseptic hip and knee implants, including Pseudomonas. Finally, we found compelling evidence for the existence of these communities. Specifically, the patient sample showed higher Shannon diversity than the open-air controls, and several taxa were significantly differentially abundant between these two groups. The open-air control showed a higher abundance of those same known laboratory and sequencing contaminants, as well as the positive control taxa. This emphasizes the effectiveness of the source tracker algorithm in selecting relatively contaminant and artifact-free samples. The patient sample showed higher abundance of a few taxa that might plausibly be part of the polymicrobial communities of presumed aseptic hip and knee implants. Taking these results together, we demonstrate that 16S rRNA gene sequencing is not ideally suited to these samples. Their low microbial abundance amplifies the effects of artifacts and differences in DNA extraction methodology. On the other hand, we show that there likely do exist polymicrobial communities in presumed aseptic hip and knee implants. The taxa we highlight should be the subject of further investigation. Thank you so much for listening.